introduce Renee, give you a little bit of her background. Dr. Renee Howell is currently Senior Director of Clinical Affairs with Siemens Healthineers, where she leads the Clinical Studies Unit for the company's Immunoassay Group. Renee has over 20 years experience in industry and has held positions of increasing responsibility at Canon Biomedical, Quegen, Diogen, and correct me if I'm saying these wrong, I apologize, and
That's okay.
Seracare. Dr. Howell completed doctoral studies at Rutgers University, New Jersey, studying in vivo genetic variation of HIV-1 and postdoctoral studies at the National Institutes of Health in Bethesda, Maryland, which focused on DNA structure and its effects on gene expression. She is currently the chairperson for the industry division of American Association of Clinical Chemistry and serves as a Siemens representative to the Joint Committee for Traceability in Laboratory Medicine and is an advisor on the Clinical and Laboratory Standards Institute expert panel on molecular methods. She has authored or co-authored dozens of papers and professional abstracts and holds several U.S. patents. And please help me welcome Renee Howell.
Thank you, Elaine and Kaidi, and thanks everybody for dialing in. I have to say that when the COVID-19 pandemic started, it really gave people who work in laboratory sciences a moment. We realized that this was really an opportunity for us to get out and do some education on lab testing, and so that was one of the reasons I thought that it might be good to do this with the library, especially since the testing has been very interesting, to say the least, during this pandemic. So I'm going to start at the beginning. I was actually given this slide. Let's see. Well, first, I actually want to go over some terms. So you'll hear this a lot. The term COV or COV is a short form for coronavirus. You'll hear SARS a lot. That stands for Severe Acute Respiratory Syndrome. And SARS-CoV-2 is actually the name of the virus that's causing the current pandemic. There was one that preceded it a couple of years ago, and that was SARS-CoV. And then the name of the disease that people have after they're infected with SARS-CoV-2 is called COVID-19. So I just wanted to put those out there before we started. So I was sent this next picture from a friend who found it in National Geographic, and this is a picture of Jun Almeida. And in 1964, she was actually the first scientist to visualize the coronavirus under an electron microscope, which is this big instrument here you see in the slide. And then on the left-hand side of the slide is actually what she saw, and those are coronavirus particles. And she and another colleague actually named the coronavirus, and they named it for these halos that you can see around the virus particles using the Latin word corona, which means sun, for the virus. So what appears to be a halo here, we can see in the next slide, which is a cartoon of the virus, are actually these big spike proteins that are shown here in yellow. And those are actually what will bind onto the cells in the human respiratory tract. So viruses are very simple in structure. They're usually a protein capsule, and then inside is a nucleic acid. And so what you see here in red is the genetic material of the virus, and that's just wrapped up in a protein capsule. So what happens when a person's infected with SARS-CoV-2 is it enters into our respiratory system, and it usually does so through the mouth and the nose, which is why we're all wearing masks, but it can also get in through your eyes in a high dose. So a lot of people who are working in the hospital or in the care of people who are infected, you'll see them wearing face shields for extra protection. So basically, the virus enters our respiratory tract through those places, our eyes, nose, and mouth, and then travels down into our lungs. And actually, a lot of the cells in our lungs, and in particular in the lower part of the lungs, are what the virus binds to. So when doctors started to look at digital radiographs of people's lungs, they would see a lot of cloud in the lower lungs because that's where the virus is attaching to and starting to cause disease. So what I really wanted to talk to tonight about tonight were the SARS-CoV-2 tests, and they basically come in three forms. There's a molecular test and an antibody test and what we call point of care test. And when you think about a point of care test, that is the kind of test that you will sometimes have done in your doctor's office, like some flu tests or rapid tests, especially at pediatricians. Another test is a pregnancy test. So there's strip tests that you can do right in the doctor's office. So for the molecular test, this is the one that you've heard probably the most about because it was the first one that became frequently used to diagnose the virus, because this test actually tests for the presence of the actual virus. And so we would see it was not a blood test. People who were collecting samples were taking swabs and sticking them up people's nose, actually right all the way to the back of your throat. So anybody who's had this test knows it's pretty uncomfortable. But what they're trying to do is get on the cotton swab cells that are infected with the virus. And then in the laboratory, that swab is put into a solution where the virus and the cells can be freed from the swab. And then that solution is used to run a molecular test, which is referred to as reverse transcriptase polymerase chain reaction. And that's done in a machine that does cycling through heating and cooling steps, which is how the virus is detected. So in a very basic form, what happens is this buffer here will release the virus genetic material from the virus particle. So it breaks the particle down, releases the genetic material, and then there's a series of materials in the test that can actually copy the virus target. So we like to think of it as a Xerox machine, for those of us who remember, or a copy machine, because we can take one copy, if it's there, and we use chemicals to just make multiple copies of it. And eventually we have enough copies to be able to detect it. So you can see with this little orange color here, where part of that reaction is tagging all of these copies that we make in a way that we can detect them later. So that's the basics of the molecular test for the virus. An antibody test, when we talk about those, that really determines if you have had an immune response to the virus. So it doesn't tell you necessarily if you're infected at that moment, but it tells you that you've seen the virus. So to talk about an antibody test, we need to talk a little bit about what our immune system does when it sees a virus or any kind of invader, which can be a bacteria. Sometimes allergens act as invaders. And what happens is when our immune system, cells from our immune system, and they're all over our body, see something like that, they can actually kind of scan the invader and use that information to produce antibodies. So you'll see here, there's 3 different cells that are capable of looking at different parts of this. Let's say it's a virus, and then they can produce different antibodies to different parts of the virus. So we call that a polyclonal antibody response, because we can make many different types of antibodies to many different parts of whatever we see as a foreign body. For the coronavirus, what's really important, as I had mentioned before, are these spike structures, which the virus uses to adhere to a human cell when it's in the respiratory tract. So what's really important about antibodies made to the spike is that they are protective, because they will find the virus and bind to those sites so that it can't bind to our cells. So while we make many, many types of antibodies, what we really want to know is not only have we seen the virus, but will we be protected from it in a subsequent round if we're exposed to the virus again. In addition to that, the immune system actually goes through a priming step. So what we see in this graph here is actually on the bottom, on the x-axis here, the days past infection. And then on the y-axis, or the horizontal axis, we see, or the vertical axis, we see a level of response. So if we follow each of these lines, the blue line here shows when you're infected with the virus. So, and then the virus, the amount of virus that we have in our body. So for a while, when you are initially infected, you don't have any symptoms, but the virus is starting to replicate. And then say at about day 5 with a coronavirus infection, you see the onset of symptoms. And after that, it becomes detectable by the molecular test. At the beginning, when you're when the virus is starting to replicate, your immune system sees that, and about day 7 will begin to produce a certain type of antibody called IgM, and we'll talk a little bit about that later. But the IgM antibody, that type of antibody, is the first one that's produced when you encounter some foreign body, and that is usually a weak response. So you see here the green line is what's showing the IgM antibody response. And then just as you are about to recover, if you have what I would say would be an uneventful infection, your body starts to produce IgG antibodies. So it's almost like your immune system in this way is primed with IgM antibodies, and then it starts to make IgG antibodies, and it's a very strong response. So this is basically the course of what they're talking about on the news about antibody testing. So there's all different kinds of antibody tests. So it's important to know what kind of test you get and what it can tell you. It can tell you if you are have IgM antibodies and you're in the beginning of the infection and you might still be infectious, or it can tell you if you have IgG antibodies, which means that you're probably not infectious anymore, but you definitely have been exposed to the virus. So this is just another slide, and I put it in here because there is another type of antibody that's important in this infection, and that's an IgA antibody. And there are tests out there for IgA antibodies as well. So IgA antibodies get made and they travel to our mucus lining. So they'll be prevalent in the nose and in your respiratory tract. And so they're very important for the kind of viruses that enter into your respiratory tract. And again, you can see in this line, another priming event in your immune system is your first response to the virus is very low, but then on subsequent exposure, you have a very strong immune response. So that's also too what we try to mimic when you're given a vaccine, right? You get a little dose so that your body can identify it and start to be primed, so that if you ever see the actual pathogen that's causing the problem, you would have a very strong response. So with that background, this is how an antibody test works. Over here in this part of the slide, on the left-hand side on the top, it shows IgG and IgA antibodies, which are just these structures that look like Ys, and then an IgM antibody, which is this big kind of conglomerate, what looks like a conglomerate of antibodies. And those are, if we have been exposed to the virus in some way, we'll have those antibodies in our blood. So they'll collect blood, separate it out, and take the serum or plasma, and then they'll react it with some kind of a solid support, usually a particle, that has engineered proteins on it that replicate some part of the protein coat of the virus. So that's what's shown here in this round object. And then we also will add some kind of chemical that will enable us to detect if we make a bridge, which is shown down here on the bottom left-hand side. So if there are antibodies in a patient's serum, they would bind with the particle, and they would also bind with the chemical that's going to produce a signal. And so we wash all that away, so we get rid of all of the signal that didn't bind, all of any other kind of antibodies you might have in your blood that didn't bind to this specific particle, and then we're able to get a reading from that. So only if you have antibodies in your system would they react in this test. Now this test is showing the kind of test that would pick up any antibody. So if there would be a little bit of a difference if we were going to go in and just try to pick up an IgG or an IgM antibody, but that's the basics of how it works. And then the last kind of tests, which we see are coming out now, are point of care tests. We call them point of care tests because they are done at the site of the patient, right? So they're not really sent off usually to a lab. And you can actually make a point of care test. It could be anything. It could be a molecular test, as I had mentioned before. In that case, it's not the little strip test, but it's a small test that's then put into a very small machine that can be run near the patient. There's also tests for antigen, which that would be a test that would test for the outside of the virus particle. So this molecular test, again, tests for the genetic material of the virus. The antigen test would test for the outside, and then antibody tests, which we just talked about. So for the point of care test, there was one that was just released actually by Rutgers University, and in that case, it uses saliva. And similar to what you've seen from the kind of genetic tests that are put out on the commercial market for Ancestry.com and 23andMe, you can use your saliva because it has cells, and it might, in the case of this virus, have infected cells. So you can use saliva to detect either the virus or antibodies. Another type of point of care test is a blood test, and it's usually done by finger stick. You can also take a blood drawn from somebody taking it from a needle in your arm, but the most popular ones are finger stick tests. And usually, just as a type of test, these tests are less accurate for a couple reasons. One is because you can't put as much material onto them as you could if you were doing a blood test. And secondly, when you're using a sample like a finger stick, there's a lot of like junk on your finger and a lot of different materials that are right in the skin of your finger, and those can sometimes interfere with the test. So they're not usually as accurate. So one of the things I also wanted to just mention is the cost of tests. So I did a little search and found out that the average molecular test is about $195. And then overall, the cost for all the different kind of tests, both molecular and antibody, range from $50 to $200. Usually the antibody tests are a lot cheaper. And we'll talk a little bit about why that is later. And from what I also understand from people who've actually gotten the test done, as well as again reading articles on it, most insurance companies are covering both tests. So that's good news for everybody. So another thing I wanted to bring up was why there were no tests in March when we saw the pandemic start, because I think it's a really important thing that I don't necessarily know if everyone has truly understood why that is. So as of today, the US now has more confirmed coronavirus cases than anywhere else in the world. So I think it is suffice to say that we were really not prepared for this pandemic at all. Usually when we're faced with a threatened infectious disease, the first line of defense we have in the country is the Centers for Disease Control. And usually part of what they do is they try to get a test designed and out very quickly to big laboratories, state laboratories, so that they can get those laboratories ready to be the first line of fight in testing. So in doing that, we had a hiccup, which was part of the materials that the CDC sent out were contaminated, so they had to pull them back and put out new material. And we realized, as we saw the rate of infection with this virus, that they were going to be too few available for what was needed. So while the CDC is a really great organization, they are not used to manufacturing at the scale that we needed for this particular pandemic. And so, we saw the White House turn to inviting industry to the White House, and I think industry actually did a pretty good job of responding once they got started. But we quickly experienced issues with raw material shortages, because it was not only the US, but all over the world that was needing tests. So every company across the world was looking for the same materials at the same time, and that really created quite a bottleneck. In addition to that, in talking to colleagues in laboratories in the United States, once we got hands on the tests, in particular the molecular tests, there was a bottleneck of the number of instruments that were available and set up in laboratories. And there was another bottleneck in the number of personnel. So there were all of these, cascading bottlenecks that were just kind of amazing to see. So another thing that the government did, which was very good, was the FDA immediately opened up their what they call their emergency use authorization process. So that allows industry an expedited path through the FDA for any kind of test that they want to introduce to the public. And I can tell you, in the course of the company I work for submitting tests, the FDA has been emailing everyone back on the weekend with questions. So they're working around the clock. But the central problem was for the United States, at least from my opinion, is that there was no coordination of the lab preparedness at the national level, right? So you can invite industry in, industry can make tests, but industry would not be able to coordinate an effort needed by a country. So we are in a situation in almost all of March, and then I think things started to get a little bit better in April, because labs had access to tests then, but they didn't have enough instruments or trained operators. So for molecular tests, most of these tests are still very manual and labor intense, so they take a long time. And the instruments that they're run on are low throughput, so you can run 100 tests per shift, for example, and they require, more highly trained personnel to run them. So a lot of the kind of tests that we use these instruments for are tests that are not emergency tests. So again, that's why the instruments haven't been designed for high throughput. So with having a molecular test as our first line of identifying people who are infected, we were in a deficit situation, since we wouldn't even have had enough instruments to throw them into labs, and then all of the personnel that are needed to get an instrument outfitted in a laboratory and working, and then having enough people trained ready to run these instruments. For the antibody tests, we're in a much better situation, since most of the tests are automated, where operators just take a tube of blood, put it on the instrument, and the instrument does everything else. Those kind of instruments that you would see in big labs like LabCorp or Quest are also high throughput, so they can handle 1,000 tests per shift, and you have these things running all day. You can get 4,000 to 5,000 results per day from an instrument. So it's a very different situation than the molecular tests. And because they're very simple to operate, they don't require the same kind of training for the personnel. So for antibody tests, which we'll now be using to understand where we are in terms of immunity for our population, we should be in a much better situation. And then I thought the last thing I would just wanted to bring up was a contrasting example of another infectious threat that we had recently, where I felt like we really were prepared. And, you know, as the Zika virus was breaking out across the world, United States first became seriously aware of it and a threat to our country because we saw in the blood banks in Puerto Rico, we started to see positives coming through the blood bank. So we knew that people were getting infected at a fairly high rate. So because it was maybe you know, part of our country and our data, we felt that we could take it very seriously and did. You know, another thing that was different about Zika is that it was transmitted by a mosquito. So in this slide, I'm showing a map down here of the United States, and you can see where the yellow shading is there, the two species of mosquitoes, their range. And so, you know, we were concerned most about protecting the borders and then protecting the borders of the states that kept warm, because mosquitoes breed, as you know, only you know, under a certain temperature. So the government did a really good job of using insecticide along the borders, in particular of Texas and Florida. And so it really prevented you know, anyone getting or any of the mosquito population getting enough of the virus dose into the human population. And then for lab preparedness, again, it was a very different situation. But there's a group in the government, a very small group called the Biomedical Advanced Research and Development Authority. For short, we call them BARDA. And what their job is to help acquire technologies and bank up supplies in the event of an epidemic or a pandemic, as we saw with COVID. And at the time of Zika, BARDA was ahead of it, you know, because they were tied into what was going on in Puerto Rico. And they actually contracted a lot of companies to design tests and design and prepare new therapies in advance. So in this case, we had everything we needed to be protected, but the epidemic never came. And that's a good thing. But I just thought it was very interesting in contrast, because it's such a different situation from what we see with COVID-19. And that really, I think, has a lot to do with the fact that COVID-19 is a fairly infectious virus, and it's spread from human to human. And it's a lot harder to control humans than mosquitoes, as it turns out. So that was what I planned to cover. And Heidi, if you want to tell me if there's any questions or anybody wants to ask any questions, I'd be happy to answer them.
Right now, there's no question yet. So if anyone wants to ask question, you can either type in the chat box, or you can just unmute your speakers and then you can speak to Renee directly.
Renee, this is Julie. I wanted to ask about your opinion on sensitivity and specificity of the antibody tests, and what is a acceptable level that we should be looking for if we're looking to, you know, get tested.
Yeah, the government, you know, the FDA is requiring us, any manufacturer who puts a test on now, it has to be 99% specific and 99% sensitive. And the reason for that is because of the, you know, relatively low infectious rate. We want to be able to, you know, not have a lot of false positives. So they're requiring very high sensitivity and specificity. And I can say that most companies are meeting that. So that's good. Any other questions?
Thanks.
Okay, Julie,
Renee?
I'm going to mute you.
Renee?
Okay, and okay.
Sure. Yeah.
So Lauren, sorry, I have 3 questions in the chat box. I'm going to read them to Renee. So if you had antibodies, would you have to take the vaccine?
So one of the other things about coronavirus is it usually has a short immunity, so only for a couple years. So we don't know what we have with the SARS-CoV-2 virus yet, but because of that, and because viruses like coronavirus change often, it might be a good thing to get a vaccine. We'll have to see. We don't know enough yet about the natural properties of the virus really to tell. But just because you have immunity doesn't necessarily mean you shouldn't get a vaccine.
And the second question is do you recommend taking the test, the COVID-19 test?
Yeah. So I think you have to ask yourself what you want to know, right? And then you have to match it up with the right test. So we don't know for sure yet what part of the virus is going to provide immunity. So a lot of places right now are doing the kind of tests that they need to do to understand what kind of antibodies will give immunity. So as I said, it's likely that the one that's used to bind onto our cells is probably the one that's going to provide protection. So we'll see if that pans out in research. But so right now we really couldn't answer that question, right? Because we have to know for sure what kind of antibodies are going to be protective. So the best that you can hope for now by getting a test is to know if you have seen the virus.
The next question is that how accurate is the antibody test at this time?
So there is some variation with what the FDA has let out on the market. The lowest sensitivity I've seen is somewhere in the high 80s, so 88%. And if you go on the FDA website, you can see all of the characteristics of the tests that have been released. But as I said, starting at the end of March, the different groups that had submitted tests to the FDA had done such a good job that the FDA raises the bar. And that's how that emergency use process goes. So different companies keep submitting to the FDA, and as the data gets better, then the FDA requires everybody to come up to that standard. So most everything that the FDA will let through will be very high sensitivity and specificity in the high 90s.
So the next question will be, do you recommend we take any of these tests once they are available to the public? If so, which test?
Again, it really depends on what you want to know from the test. If you suspect you had an infection and you want to know, the antibody tests will probably tell you that. If you feel bad and that's when you should get the molecular test. Go to your doctor, and the doctors are pretty aware now of what the symptoms are, especially unfortunately in our area, because they've seen so many cases.
Yeah, okay. Before we go on, I think Lorraine has a question to ask. So let's ask, Lorraine, you can ask the question first. Lorraine?
Yeah, can you hear me?
Yeah, hi Lorraine.
Hi, how are you doing? My question is really kind of a strange one, but would it be possible to ever give a talk on how does the virus come to be? I've been trying to find information where, how, why?
So it's a really great question, Lorraine, and I've seen some of this in the news. And again, if I know it's hard on the internet to know what's real and what's not real,
Right.
but maybe I could send some sites on that, because what is happening, just like Zika, what's happening with coronavirus is there are viruses that are in animal and insect populations that have been around for a long time, but we've never gotten exposed to them, and it's been some change in the environment that caused transmission to happen, from an animal to an animal or an animal to a human. So
Do we know why a virus happens? I mean, it had to come from someplace.
well, viruses have been around as long as we have.
Mm-hmm.
So it's not like it just didn't appear. It's been around in the world for a very long time, but it was hiding in some animal population. In this case, they suspect bats, and then there was a series of circumstances that led to a bat infecting another animal, and that animal had closer contact with human population in Asia, and that's what happened.
Okay, I'd appreciate getting those sites.
Yep.
Thank you. Thank
Okay.
you so much.
So, the other part to that, Lorraine, is a lot of these changes in the way humans interact with animals or insects is related a lot to changes in climate. So, as that happens, it disturbs kind of the existing order of things, and puts two species together that haven't been together before.
Thank you.
Yep. Any other questions?
So I'm going to continue with the next one. From chat box, can the drug losartan be effective against COVID?
I don't know off the top of my head what kind of a drug that is, but I can tell you there's, there were, at the very beginning of this, there were kind of non-specific drugs. One was a treatment for malaria that people thought about. And the reason people were thinking that it might help is a lot of those kind of drugs will react just non-specifically to dampen immune response. So one of the things that happens with COVID is if your immune system isn't able to resolve the infection, we're seeing a lot of people go into a kind of hyperimmune response. So they call this, if you've seen it in the news, the cytokine storm. But what happens in some people, and in particular, people who are 60 or older, is your immune systems become dysregulated when you get this infection. And so a lot of the drugs are that we're looking at are related to suppressing the immune system. And it's a tricky thing because you need your immune system to react to this virus to clear it. But if it goes, out of whack, which is what is happening, then you need to really suppress the immune response because it can be such a strong thing, it'll kill you. So there's those kind of drugs. There's also drugs that are antiviral, and there's been some success with them. Now they're more specific because they're going directly to suppress the virus. So those are the two kind of drugs that I see out there right now, things that are non-specific and things that are very specific to the virus. The best thing would be a vaccine.
Okay. Okay, the next question is, have the treatments with blood plasma been successful?
Some have. Very early in February, there were reports coming out in the scientific literature where they had tried this in Wuhan, and they had, a pretty good success rate. And this is a therapy that's been used for, again, a very long, long time. Is once you have somebody who's convalesced, they've gotten through an infection, they probably have antibodies, then their blood becomes very valuable because it can be used to treat somebody who's sick. So, it's not a guarantee to work, but it can help in some cases.
If a person tested positive for antibodies, how long the person is contagious?
So for corona, how long is the person contagious?
Contagious, yes.
Okay. So I'm going to see if I can go back to that graph that shows you, because usually it depends on the antibody that you're looking at. So if we go back to this graph, you'll see there is a time when you have both antibody and virus present, and so you're still contagious. So what you really are looking for is that you have an IgG antibody response and that you are free of symptoms, and that gives you a fairly good confidence that you're not infectious anymore. But coronavirus seems to, in some people, stay around for 28 days. So people can shed that virus for 28 days.
Okay, the next question is, does the amount of antibodies correlated to the how sick someone got when they had COVID-19?
So that's a complicated question. None of these tests quantify the antibody response because it doesn't necessarily correlate with the kind of infection you had, whether you had a heavy viral load or a light viral load. There's a lot of genetics behind your immune system too. So sometimes people are just naturally strong responders no matter what kind of dose they get. So it's useful for research to quantitate antibodies as you're trying to work out what the natural behavior of the virus is, but for a diagnostic test, it's not very helpful because it doesn't tell you the kind of information that you're asking about.
And I saw Omara is unmute herself. Do you have a question?
Yeah, I have a quick question. Renee, could you tell us why there are some people that are asymptomatic and others develop symptoms?
So, people who are asymptomatic have a very mild infection, but their immune response is enough to get rid of the virus. So like I said, it's a very there's a lot of genetics in that. There's a lot of things that would suppress a person's immune system. So like as we age, your immune system gets depressed. People who smoke or people who have underlying diseases, all of that kind of thing suppresses your immune response. But I think what we'll see is, for the most case, there's a lot of healthy people who've probably had the virus but don't know it because they've been asymptomatic. So that's what a lot of this testing will help us work out. One study we're working on now is looking at healthcare workers as they, samples were collected through, starting sometime in March, and we're looking at were any of the healthcare workers asymptomatic carriers? So we're trying to understand that piece of it too.
So Omara, you have other question to ask?
Nope, that's it.
Good. Okay. So the next question is that how long after exposure to COVID-19 is one able to start infecting others?
So basically, as soon as you have the virus, you're capable of shedding it, and it replicates to a certain amount, you're capable of shedding it. So, even after you see on this graph, 5 days is when you start to see symptoms. So even before that 5 days, you could be spreading the infection. So that's why it was really, important to sequester people who had traveled, even if they felt okay, to make sure that they, if they were asymptomatic, they still weren't shedding the virus and spreading it.
I think the last question will be I'm sorry, not the last question. The next question is that how long is the virus airborne?
So there's been a lot out there on that, and I just don't know off the top of my head. But it's long enough when you're out and about and walking that you really want to wear a mask, because even if you just walked by, or somebody just walked ahead of you for a couple minutes, there still might be aerosol around.
Sorry, I skipped a question earlier. Why has the virus caused blood clots in some people? So
that's a great question. So that's part of what happens when your immune system kind of goes out of control. One of the effects is that your clotting system is activated as part of a non-specific immune response. And And again, that usually is resolved in most healthy people, but in certain populations, in people who have underlying diseases, your coagulation system just goes out of regulation and starts clotting all throughout your body. So again, that's part of your immune response.
Do you think at some point that nearly everyone should get tested in one form or another?
I think we have to have a certain amount of the public tested to understand, where we are with having the population immune in the case that we have a second wave of this. So I don't know that everybody has to be tested, but we have to understand as a country, what we need to do on a vaccination front. And so there we have to understand what the general immunity level is.
Does the coronavirus shed only from the nose, mouth? Can it shed from the skin?
Not from the skin, from mouth, tears, any kind of fluids from your nose, and it can be found in your blood. If you get a really large infection. So we don't know about blood transmission right now. There hasn't been any demonstrated case of it, but that's something that people are looking at as well.
I saw Julie unmute yourself. Do you want to have a question to ask?
Yeah, just following up on this graph. So does the antibody test tell you where you are? If you're positive, this is like, for example, like if you're in the IgM stage, you wouldn't be tested positive for the IgG, so which means you're more contagious at that stage, right?
So you could have both IgM and IgG at the same time. And so again, it depends on what kind of test you got. There's tests that will tell you, they're called total antibody tests, so they're not going to distinguish between what type you have. There are tests specifically for IgM and IgG. So if you got, say, the test for IgM and IgG and only IgM was positive, you can assume that you're, at the beginning stage of an immune response and you're likely still infectious.
Right.
You really have to put the information together.
Okay. I didn't know there were actually tests that didn't offer both. I assume they were testing for both, but you're saying they don't.
So no, it's very dependent on the manufacturer. So you should ask when you're getting a test, is it specific for one type of immunoglobulin, or is it going to pick up all of them?
Got it. Thank you.
And back to that other question, is it spread on your skin? If you cough or if you get, saliva on your skin, then you can spread it that way. So I just wanted to make sure people understood that. That's why, again, they're saying don't touch your eyes, don't touch your mouth, don't touch your nose, and wash your hands a lot.
Okay, the next question. Can there be herd immunity even without a vaccine?
So herd immunity requires a lot of people to have seen the infection. So, while that's possible, I think the only way we're going to get it is with a vaccine.
Why do you think it's important to wear a mask while walking or running outside? For example, on the Bronx River Parkway path, if you are keeping at least 6 feet away from other people.
Yeah, so if you're 6 feet away from other people, you are probably, safe. So a mask becomes an extra protection. But if you're going to get closer to people than 6 feet, you're still in that aerosol cloud. So you really want to wear a mask and prevent that those potential particles from getting into your respiratory system.
I think I hope I didn't skip any of your questions. If I skipped, please feel free to unmute yourself and speak to Renee directly. Or if you have any other question, please unmute yourself. You can speak to her as well. Okay.
So Renee, can you tell us why it seems that the virus, that more people of color are impacted by the virus?
So from what I have read, I would say the biggest contributing factor to that are, socioeconomic reasons. So more African American people tend to live in denser places, tend to not have as good access to healthcare, and that's really the predominant driver of that. There can be all kinds of differences between people based on genetics, but I think the real driver of that are more socioeconomic causes.
Anyone have any questions? The other question I skipped earlier is that I think some patrons wants to know if the recording can be available to after the program.
Yeah, usually you guys do post the,
Yes.
right, it's recorded and it's posted.
Yeah.
Yeah. So Renee, if you are fine with it, we can post it.
Yeah, I'm just going to go to my disclaimer at the end so folks know that these are just my views, that I'm expressing, my professional views. And to remind everybody that really, when you get tested for anything, you really need to do that with your physician, because it's important to understand test results in the context of your medical history. And
there's a question that was missed. It says, do you think it's safe to go in an elevator with other people, I guess?
If that elevator is really, large, more than 6 feet. But I wouldn't go into an elevator without a mask.
And Mike Dardano says, well done, Renee. Thank you for all the information.
Thanks, Mike.
Okay.
When will this be available that we can see it online at the library?
I think as long as we finish all the editing, and we will send, I think Elaine is still there. Can you
Yes,
answer
I
the
am.
question?
So we are the tech crew, Heidi and I. And so we are editing. It's probably going to be about 10 days.
Okay.
Because we're still working on some of the other ones, but we are getting more adept at it. And then Tuckahoe Access gets them up pretty quickly.
Okay. Thank you.
Thank you.
Renee.
Thank you, Renee.
There's one more question from Virginia. Will the coronavirus vaccine be separate from the regular flu shot?
Ah, that's interesting. I assume it will, but we'll see. What creative solutions people come up with.
And Cheryl says, excellent presentation. Thank you so much.
Thanks.
Thank you, everyone, for
Thank you,
your
Renee.
excellent questions.
Thank you,
I think the question
Thank you,
to
Renee.
Thank you so much, Renee.
Thank you, Renee. It was awesome.
All right. Thanks.
Thank you for
Thanks,
sharing
Renee.
expertise. Thank you.
Thanks for coming out.
Thank you, everyone.